Um, Also, thing to note is high oxygen inhibits glycolysis, whereas low oxygen encourages glycolysis. Because glycolysis is an anaerobic step, it makes sense to happen in low oxygen. But when there's high oxygen, we actually don't need glycolysis as much because we get a lot of energy from the other pathways we're going to learn in the future. Just checking how I am on time. I'm going to end this through the other pathways that happen with pyruvate. So when we turn into pyruvate, which is the final product of glycolysis, there's a lot of things that can happen to pyruvate. The first thing is we can turn it into lactate. And so the reason why we want to turn it to lactate is to recuperate the NAD. So you can imagine that the product of glycolysis is NADH. But NADH, to kind of provide a preview, need oxygen to use NADH for energy. And if you are in an environment where NADH can't be used for energy because there's no oxygen, you need to get rid of this NADH. That way you can use more of the NAD for glycolysis. And so for us, mostly, the way we do it is we transfer the electrons in NADH to pyruvate to turn it to lactate. And so this reaction is an oxidation reduction because the pyruvate goes from carbonyl to alcohol, which is a reduction. And then the NADH turns into NAD, which is an oxidation. And if you're not surprised, the name of that enzyme is dehydrogenase. So in this case, it's lactate dehydrogenase. And there's this thing in the cell, in the body called the Cori cycle to kind of explain why this makes sense. So in the muscle, we turn glucose into pyruvate. And if you guys remember, that's literally what we talked about this recitation, that's glycolysis. But we want to regenerate the NAD. So we turn pyruvate to lactate. Now the lactate seems useless in the muscle. And so in order to make that lactate usable again, we send that lactate to the liver. And in the liver, we'll turn that back into glucose. And we'll talk about this pathway soon. But the lactate will turn the liver will turn the lactate back to glucose, which will be usable again in the muscle. So this kind of cycle of glucose to lactate to glucose again is called the Cori cycle. The alternative for some microbacteria is ethanol formation. And in this step, the goal is the same. It is to regenerate the NAD. But in this reaction, it's a little different. So the first thing that happens is we actually form acetaldehyde. So we go from pyruvate to acetaldehyde. And the one thing to note is first, we keep this part of the molecule the same pretty much, but we lose this COO group, and now it's turned into an H. So this reaction right here, because it gets rid of a carbon dioxide, or if you think like a functional group, it gets rid of a carboxyl group. This is called a decarboxylase reaction. So this step right here, where you get rid of CO2, is a decarboxylate reaction. And this is involving these three cofactors, TPP and Mg2+. So the way it works, there is this carbon ion, TPP. You kind of just have to memorize that this is what the carbon ion looks like. But essentially, in this conformation, the carbon is a nucleophile, and it can attack the carbonyl. Once it attacks the carbonyl, we form an OH, and then we form, and then the carboxyl group is visible on this side. What's special about this is that if we transfer the double bond to the nitrogen, it allows for this CO2 group to leave. And so this first step right here is nucleophilic attack. Then the second step is the CO2 leaves, and we generate this group, which I hope you guys recognize is an enol. And then this enol group, we can allow it to leave the TPP once again through a series of steps like this, where we first generate a carbon ion again by having the lone pair come back. And then after we generate the carbon ion, we have an H plus attack that. So now we have an alcohol group. Then this alcohol group can leave by reforming a carbonyl. If the electron pushing to you doesn't make sense, I hope that without my annotations, it might be a little more clear. But feel free to rewatch my explanation if that helps. But essentially, there's two steps to this fermentation in comparison to the lactic acid fermentation. The first part is we get rid of CO2 using TPP Mg2+. And then the second part is the actual oxidation, where we turn this aldehyde group into an ethanol by having NADH give its electrons to acetaldehyde. So NADH gets oxidized, whereas ethanol gets reduced. This question is a favorite in asking for mechanism. So it's almost often that they'll ask you in ethanol fermentation, what is the most important cofactor? You should remember it's TPP, thiamine pyrophosphate, which is in this case, this molecule right here. So I would highly recommend that you write this on your cheat sheet. It's a very hard structure to memorize, but if you should remember what it does, thiamine pyrophosphate is a nucleophile. And it's not only is it a nucleophile, it's a nucleophile that once attached allows for CO2 to leave. So that's how you should remember TPP. It's a nucleophile that allows for CO2 to leave. Because we're running out of time, gluconeogenesis is a very complicated pathway. I will cover it in the next recitation, but if I were to provide a preview on this, you should think of this as the reverse of glycolysis, almost. If glycolysis is glucose to pyruvate, so let me write that here. If glycolysis is glucose to pyruvate, gluconeogenesis is the reverse of the pathway. And you might be thinking that the reverse of glycolysis is something related to photosynthesis, because generally gluconeogenesis is not talked about in high school or in intro bio, but this is actually very different from that. So this is not something that is done by plants. This is mostly done by us, especially in the liver. And so what I hope you guys appreciate before we start is that one, the pyruvate turns into glucose in gluconeogenesis. So in this drawing right here, the glycolysis step is on the left, and then the gluconeogenesis step is on the right. The other thing to note is that glycolysis runs this way, glucose to pyruvate, whereas gluconeogenesis runs upward, pyruvate to glucose. And the last thing to note is that in these two pathways, a lot of the enzymes are the same. So in all these parts that are not red boxed, the enzymes are the same, but in some steps, there's a difference. In some steps, we see certain steps are different in comparison to the two pathways. And to a lot of people's surprise, it actually is these three steps. The reason why these three need a different enzyme is because these are the three irreversible steps of glycolysis. So we'll cover that in depth in the next recitation. I'll start with that in the next recitation. And we'll go over the pathways for those, which involve this mechanism. But to kind of leave you so that you can kind of understand sugars and hopefully to kind of guide you on the coming up lectures for Professor van der Heijden, we basically talked about glycolysis turning glucose to pyruvate. And so by now, where we are in the pathway is we have pyruvate. And so in the next coming lectures, like today and tomorrow, Wednesday, we're going to learn how to turn pyruvate to acetyl-CoA, and then turning that acetyl-CoA into carbon dioxide. And then overall, while we talk about all of those pathways, We are generating a bunch of NADH. So then the next couple lectures, we'll also talk about how that is metabolized. And also to kind of give you a full perspective so you don't just memorize this out of the blue, we will also talk about how other molecules like amino acids and fatty acids are consumed so that we can actually use it in this pathway. Okay, it's 2:55, so I will end my recitation there. I will try to cover gluconeogenesis and the TCA cycle next recitation. But for those of you who are in the recitation, do you guys have any questions? Okay, for that, no questions, then I'll see you guys next week.
Thank you.
Whoa, this is really weird.
I know I have to edit it. Why do you